Hello, biology buddies. I hope you, your family, and friends are safe and well. This is lesson 4 from Unit B1 Cell Biology, and this lesson is all about standard form, the SI units, specifically their prefixes, and the sizes of cells. So in previous lessons, lesson 1 and lesson 2, we looked at plant, animal, and bacterial cells and the different organelles or structures that they have within them. For this lesson, at the end, we're going to start to put some numbers to those so that we have relative ideas of how big they are. We're also going to practice using standard form to write down numbers, and we are going to make sure that we can convert between the different prefixes in the SI units. So by the end of the lesson then, you need to be able to recall the prefixes used in scientific measurements. These include millimeters, meters, kilometers, micrometers, nanometers. We're going to use those to describe the size of different cells and organelles. We're going to convert measurements from one unit to another, so from millimeters to meters, from nanometers to millimeters, etc. And we're going to practice using standard form when we're describing the sizes of things. Let's get started. Standard form. Standard form is really useful in science. It's going to come up in biology, it's going to come up in chemistry, it's going to come up in physics. It's one of those things that you are going to have to master and be able to interpret, but also record answers in. Standard form will always look like a times 10 to the power n, where a is a number between 1 and 10, so it's any number bigger than 1 and less than 10, and n can be any positive or negative integer. So for example, 3.2 times 10 to the 3 is standard form. 10 to the 3 means 3 zeros, so times 1,000. It also means you move the decimal place 3 times. So here I would move this once, twice, 3 times. So that is going to be 3,200. If I were to write this as 32 times 10 to the 2, that's also 3,200 or 3,200, but that's not standard form. And the reason that it's not standard form is that that first number there needs to be a number bigger than 1 and less than 10. So obviously 32 is bigger than 10. That's not going to work. You can have negative numbers. So for example, we could have 3.21 times 10 to the minus 1. That's fine, that's still standard form. What that means, 10 to the minus 1, is that you are dividing by 10. So you're going to end up with 0.321. If you wanted to write that in standard form, it would be 3.21 times 10 to the 1. So a couple of different ways of doing it. One is convert your answer into the full number. So 10 to the 3 would be 1,000. 10 to the minus 1 would be 0.1. Or you can move the decimal places. So if it is a positive n, if the 10 to the power is positive, then you're going to move your decimal place to the right. If it's negative, you move it to the left. Let's have a look at a few examples. This is one of those things where if you practice it, it becomes really simple. So what is 1,739 in standard form? We need to remember that we want a times 10 to the n. That a needs to be a number bigger than 1 and less than 10. So the number that we will pick here that's bigger than 1 and less than 10 is 1.739. 1.739. That is our a value. Now we need our 10 to the power n. We move the decimal place 3 times, so it's going to be 10 to the power 3. Why is it plus 3 and not minus 3? Because we said if it is a positive, we're going to move the decimal to the right. So let's see what would happen. It's positive, so we would move the decimal 1, 2, 3. And there we go, 1.739 times 10 to the 3. Okay, next one. What is 0.00542 in standard form? First thing we need to do is figure out what a is going to be. A needs to be a number bigger than 1, less than 10. So we will pick 5.42 as our a value. This time we're going to have a negative number for n. And how many are we going to have? Well, we move the decimal place 1, 2, 3, 4 times. So it's going to be 10 to the negative 4. And I'll do that in blue just like last time. 5.42 times 10 to the negative 4. Why negative 4? Because I moved it 1, 2, 3, 4 times to the right. Next question. How much is 1.34 times 10 to the 5? 1.34 times 10 to the 5. This is a positive number. If we look back, positive number, we're going to move the decimal to the right. So we have 1.34. I'm going to add some zeros on at the end here. They don't mean anything because they are after a decimal place. If you add numbers to the end of a number after the decimal place, it doesn't change anything. This is just so that we can see how many times we have to move our decimal place. So we're going to move it 5 times. So 1, 2, 3, 4, 5 times. And that gives us an answer of 134,000. So 134,000. Next question, how much is 6.34 times 10 to the minus 4? We have 6.34. This time, it's negative. I'm going to be moving the decimal to the left. So to help out with that, I'm going to put a bunch of zeros at the front. And just like before, if you put zeros before the decimal place, it doesn't change the number. So 0.6 is the same as 6. 0.000000006 is the same as 6. We're just doing that to help us figure out where we're going to move our decimal place to. So we're moving to the left this time because it's a negative number. So 1, 2, 3, 4. We're going to put our decimal place right here, which means we're going to end up with 0.0000634 as our answer. So there we've done 2 examples, one with a positive power of 10, one with a negative power of 10, for both going from a number to standard form and then going from standard form to the longer number. You'll need both of those models when you're doing the questions later on. The next bit we're going to look at is SI units. SI stands for Système International, which is the International System of Units. It is a way that scientists from all over the world can communicate with each other when they're doing measurements. So for example, everybody uses meters, everybody uses kilograms. You won't find scientific papers with things like inches, pounds, stone. Use the metric system. Really, really important that when people are writing papers and communicating with each other, that they get those measurements right. Imagine if you were going to buy something and you thought that it was in pounds, but it turned out it was actually in gold bars or something like that. It gets very confusing, it gets very messy. So it's really important that everybody has the same system of measurements. We're going to focus not so much on the units, we're going to focus on the prefixes, so the bit that goes before the unit. They have a very specific symbol and they mean essentially a number. So for example, kilo, which has the symbol of a lowercase k, means 1,000. And if we do 1,000 in standard form, it's going to be times 10 to the 3. Centi means 1/100th. Kilo, by the way, comes from the Greek. Centi and milli come from Latin by way of French. So cent in French is 100. 0.01, so a centimeter is 1/100th of a meter, which is 10 to the minus 2. Milli is 10 to the minus 3. Micro, which has this Greek letter there, is 10 to the minus 6. and nano is 10 to the minus 9, so super, super small. You'll learn quite a bit about nanotechnology and nanoparticles in a section in chemistry. And if you're doing physics, you're going to go to prefixes that are bigger than kilo. So you might have mega, you might have tera. You'll also see those when you're talking about things like computer storage space. You'll notice that this is blank here. The reason for that is for the bit that's worth 1, there is no prefix. Let's have a look at how it plays out with meters, just so you get the full word. So we have kilometer, meter, centimeter, millimeter, micrometer, nanometer. A kilometer or a kilometer is 1,000 meters. A millimeter is 1/1000th to 10 to the minus 3. A micrometer is 10 to the minus 6 meters, and a nanometer is 10 to the minus 9. What that means is that there are 1,000,000 micrometers in 1 meter. There are 1 billion nanometers in 1 meter. Really, really, really small. So those are the values that we're going to be using. Those are the units we're going to be using when we're talking about things like the size of cells and the size of organelles, because cells are crazy small, so we need to use units that are crazy small. Whenever you're doing a question in biology, one of the first things that you want to check, and we'll have a go at this next lesson with some microscopes questions, is you want to check and make sure that the units are consistent the whole way through. Often they'll add an extra mark to a question on an exam where you have to convert from one unit to another. So for example, they might give you one value in millimeters and another value in micrometers. They might give you one in nanometers and micrometers, and you need to make sure that the units are all the same before you start to do your calculations. Let's look at some examples of how to do that. So we've got convert meters to millimeters, micrometers, and kilometers, millimeters, nanometers. Really, really simple rule to follow here. If we go back, we're going to ignore centimeters. You'll see why in a second. There is a very, very simple rule. When you go from big to small, you are going to multiply. And when you go from small to big, you are going to divide. How much you're going to multiply and how much you're going to divide by? Well, you'll see that there's a difference in the power of 3 as you go down, and that's why we're ignoring centimeters. So when you were going from kilo to meters, you were going to multiply by 10 to the power 3. When you were going from meters to millimeters, you were going to multiply by 10 to the power 3 by 1,000. So every step you take, kilo, meter, milli, micro, nano, you're going to multiply by 1,000. Oops, wrong color. And every step you go in the opposite direction, you're going to divide by 1,000. So we can quickly add this in the top corner, and if you have trouble remembering this, I suggest that you make yourself a little chart like this in the corner of your test or exam paper. If we go this way at every step, we're going to multiply by 1,000. You'll see that I've skipped out centimeters just so that we don't get confused. And if I go in the opposite direction, I'm going to divide by 1,000. So again, that's 10 to the 3, and that's 10 to the minus 3. So convert 12 meters to millimeters. Well, I'm going to go from meters to millimeters. So I need to multiply, and I'm going one step, so I'm going to multiply by 1,000. So 12 meters times 1,000 is equal to 12,000 millimeters. How many micrometers are there in 0.03 kilometers? So again, we're going from a large number, kilo, down to a smaller one, micro. This time we're going to do 2 steps. So we are going 1, 2, 3 steps actually. So we are going to have 0.03 times first step, second step, third step. Now, another way of writing this is times 10 to the 3 times 10 to the 3 times 10 to the 3, which is going to be times 10 to the 9, which means we're going to move our decimal place 9 times, or 1,000, 1,000, 1,000 to the right. and we're going to end up with 0.03. Again, we can add zeros to the end there, it doesn't mean anything. 1, 2, 3, 4, 5, 6, 7, 8, 9. So we have 30 million. We don't want to forget our units. So 30 million micrometers. Last one, how many millimeters are there in 239,144 nanometers? For this one, pick a new color. We are going up the ladder. So we're going 1 step, 2 steps. So we're going to divide by 1,000 each time. So we have 239,144 divided by 1,000 divided by 1,000. Another way of writing that is times 10 to the minus 3 times 10 to the minus 3. Remember, 10 to the minus 3 is equal to 1 over 1,000, which is the same as divided by 1,000. Another way of writing that is times 10 to the minus 6. So we have 239,144 times 10 to the minus 6. We can add our zeros to the front. So we have our decimal place right here to begin with, and we're going to move it 6 times to the left. So 1, 2, 3, 4, 5, 6, and we end up with 0.239144 millimeters. So there are a number of different ways of doing this. The best way to do it is to use the 10 to the power, work it out that way. Don't forget your ladder. If you are confused about kilometers, millimeters, meters, nanometers, micrometers, draw that out, and remember, when you're going down the ladder, you're multiplying by 1,000. When you go up the ladder, you're dividing by 1,000. Do be careful with centimeters. You are going to, from meter to centimeter, multiply by 100. If you're going from centimeter to meter, you're going to divide by 100. And if you're going from centimeter to millimeter, you're only going multiplying and dividing by 10. Okay? But you are going to have to convert. Look out for those in the questions. And spoiler alert, you will have questions in the next lesson where you're going to have to apply this skill. Let's take a look then at some rough cell sizes. You'll notice on the x-axis here, we've got nanometers, we've got micrometers, we've got millimeters, and we've got meters. Every step along here, you are multiplying by 10. We call this a logarithmic scale. So it's not your normal x-axis. You're not going up by the same amount each time. You're actually multiplying by 10 at each time. So let's take a look at the size of some things. One of the things that I found interesting from this diagram is that an ostrich egg and a chicken egg are not actually that big of a difference, even though an ostrich is obviously going to grow up to be much, much, much bigger than a chicken. Human eggs, which are around 500 micrometers, maybe a bit less, are much, much smaller than frog eggs, chicken eggs, and ostrich eggs. That's because humans are placental mammals, while frogs, chickens, and ostriches are not mammals. So they don't keep the egg inside their body. So in that egg is going to need to be all the food that the animal is going to need to develop. We have the relative sizes of cells. You'll notice that there is that cheeky little hashed line there because animal cells and plant cells can range, depending on what type of specialized cell they are, from about 10 micrometers all the way up to 100 micrometers. Bacterial cells are very, very small. Viruses are even smaller than bacteria. So remember, we said there's more bacterial cells in your body than there are human cells. One of the reasons for that is the bacterial cells are much, much, much smaller than the animal cells that make up the rest of your body. You will also notice along the bottom here we have the idea of an electron microscope and a light microscope. We're going to talk about those in the next lesson, lesson 5, on microscopes, microscopy, and the magnification equation. What sort of question might you get that deals with these sorts of things? Something that looks like this. An average animal cell has a nucleus with a diameter of roughly 6 micrometers. Use the diagram to explain why bacteria don't have a nucleus. So if we look for the bacteria have a rough size of 1 micrometer. So if the bacteria is 1 micrometer and the nucleus is 6 micrometers, there's no way that the nucleus would fit inside the bacteria, so it's impossible for it to have it. Another common question that you'll see is, why don't bacteria have mitochondria? Again, it's mostly a size issue. The mitochondria are in many cases actually larger or about the same size as the bacteria, so they just wouldn't fit inside. Okay, let's have a look at the outcomes for the lesson. We said we were going to recall the prefixes used in scientific measurements. So kilo, centi, milli, micro, nano. Kilo is 1,000. Milli is 0.001 or 1 over 1,000, 10 to the minus 3. Micro is 10 to the minus 6. Nano is 10 to the minus 9. Describe the size of cells and organelles using appropriate scientific notation. So talking about cells using micrometers, or if you're talking about really small bits of cell, maybe even nanometers. Convert measurements to different scales. We did that. We remembered our ladder: kilometer, meter, millimeter, micrometer, nanometer. Go down the ladder, multiply by 1,000 every step, go up the ladder, divide by 1,000 every single step, and then use standard forms. We did some practice of using standard form as well. Thank you very much for listening. Like I said, next lesson will be about microscopes, and we will continue to apply some of the skills that we learned in this lesson in that lesson. Talk to you soon. Bye-bye.